Hi, everybody, and welcome to my seminar. My name is Jed Mahoney. I'm the stem cell biology team leader at the Cystic Fibrosis Foundation Labs, and I'm here today to give you an update on the iPSC-directed differentiation to the airway epithelium. I'd also like to just reiterate that I will be checking chat and I will be checking my messages, so feel free to take part in the discussion that way. Or if this is beyond the NACFC and you're seeing it for another time, feel free to email me at jmahoney@cff.org. I have no disclosures to report. So why are we talking about deriving airway epithelium from pluripotent sources? Well, really for two veins. There's the ability for iPSCs to impact drug screening because there are over 300 known to be disease-causing mutations for CF, many of which are incredibly rare. We are lacking our necessary material, which is generally primary airway epithelial cells, to be able to develop new therapeutics for some of those more rare variants of CFTR. So we could technically take an iPSC from a patient, direct the differentiation towards airway epithelial lineages or intestinal lineages, perform drug screening on that, and be able to advance our therapeutic development pipelines. But there's also another side. There is the approach that which we can take a cell from a patient, generate pluripotent iPS cells, and correct the disease-causing variant in CFTR, direct the differentiation towards the airway epithelium, to what stage we're unaware, and then utilize those cells as a transplantation for hopefully a single one-time cure for those who suffer with cystic fibrosis. And now, before I go any farther, I really find it's important that we look backwards and we respect the amount of work that went on before us. Over the last decade and plus of years, many publications have come out, starting with early patterning towards the foregut and endodermal lineages, and getting to more recently, we're seeing increasing maturity and function in the lung epithelial cells that are being derived from pluripotent sources. So basically, the take home from this slide is that we stand on the shoulders of giants, and this work has been a collaborative effort between many labs, with many individuals taking part throughout the years. And the other thing I think we need to go over before we dive in is what's the goal? Well, the goal is to generate material very similar to primary human bronchial epithelial tissue. Here, I have two immunofluorescent stains, one at day 0, which is pre-differentiation, where you can see p63 expression homogeneously across the plate. After 28 days of differentiation at an air-liquid interface, you start to be able to appreciate acendyl-alpha tubulin and plunk, markers of secretory cells and ciliated cells. This has been the gold standard in drug research over the years, and this is where many drugs have actually come through as validated for CF. But it's not just the markers that matter. It is actually the whole entire cell that matters. So what we've done is we've utilized those primary cultures, and we've performed single-cell RNA sequencing of the differentiated cells at ALI. This has given us what we would call a reference transcriptome. We can see here basal, secretory, and ciliated cells quite easily that align with the markers of the known cell types, p63 and keratin 5 for the basal cell, SCGB1A1, a club cell marker, and FOXJ1 marking the multiciliated lineage. But when we think about this, we can't just use one gene. So actually, this work was used a classification system based on the Plashart et al. Paper of 2018, correlating approximately 20 to 25 genes per cell type. If you'd like to learn more, unfortunately, I don't have time to dive into it now, but you can see that work by Plashart et al. 2018. So what does it look like to do directed differentiation experiments? Well, basically, we follow developmental markers and time points. And these milestones, we just image in the dish. So we'll watch OCK4, a marker of pluripotency, vanish as it gets replaced by SOX17, which will then turn off focally, and then NKX2.1, an early progenitor of lung marker, will activate. After that point in time, we have to mature lung to adult. So we have to get NKX2.1 to become p63, keratin 5, and NGFR positive to be like a basal cell of the airway epithelium. And those cells we asked to differentiate to both the secretory and ciliated lineage, and we observe SCGB1A1, FOXG1 activity. And what does that look like? Well, it looks like this. Here you see 3 independent iPSC cell lines that have gone through directed differentiation, and they have been triple labeled for OCT4, SOC17, and NKX2.1. And you can see early on, those pluripotent markers are ubiquitous across the slide and start to fade off as the differentiation occurs. And then you start to see the activity of SOC17. As SOC17 propagates across, it is then again lost for the NKX2.1 expression. This is what we mean by directed differentiation, activation in a sequential-like pattern in the markers that follow development. Now at that day 15 and day 11 to 15 time point, we have a co-culture in the dish. We find both intestinal and lung lineages as marked by NKX2.1 and CDX2. Here, we found, interestingly, that CFTR actually correlates with CDX2 positive cells. And if you want to know what we do with CDX2, CFTR positive cells, I ask you to go see poster 390, Megan Simpkinson. But at this point in time, it was pretty obvious that we were going to have to isolate NKX2.1 positive cells if we wanted to derive airway epithelium away from the NKX2.1 population. To do so, many labs took part. For instance, Finn Hawkins et al. In 2017 described the generation of an iPSC line that harbored an EGFP reporter in the NKX2.1 locus. This allowed for isolation of NKX2.1 progenitors from the CDX2 positive populations. We then took the same cell line and engineered M-Cherry to report on the P63 locus. This gave us a dual reporter and allowed us to be able to isolate and visualize the activity of NKX and P63 genes while doing directed differentiation. This was a major tool, which is available to anybody who would like to ask for it at cff.org/lab material requests. But this tool allowed for us to be able to generate this multi-step process to get airway epithelium. It starts with specification, as I described earlier, in a 2D format, and then moves into a 3-dimensional culture after sorting either NKX2.1 or CPM, another surrogate for the lung markers, and extracellular effaced. Those cells become proximalized and sorted again for NKX and P63 expression. And in this fate, we have a basal cell we have a basal progenitor cell, which can be continually cultured and maintain its fate, even if frozen down and thawed. One more isolation step for NGFR, NKX, and P63 triple positive cells, cells that we will term iBasal or IBC cells hereforward, are the ones that are capable for multilineage differentiation. If you'd like to know more about this protocol, I would refer you to the bioRxiv paper by Finn Hawkins described on the screen. Now, we're going to go through 3 major validations to understand, are these cultures really well differentiated? We're going to look at the markers. They should match. We're going to look at the transcriptome. When we do an unbiased analysis, they should overlap. And then we're going to ask the question that's most important for CF. Can we use these for studying CFTR function, and specifically in those rare variants that are underrepresented in the primary material for drug screening? So, pre-differentiation, we can look for the markers of those basal cells, very similar like I showed you in the 3rd slide. Here we have P63, CK5 that indicate basal-like, and NKX2.1, which indicates lung. And this matches very closely with that of the primary material that you can see has a very similar phenotype. When we ask those cells to differentiate, you can see keratin 5 and P63 remain, while other cells in the dish bring on acetyl-alpha-tubulin and in the normal multiciliated-like phenotype, MUC5A, which is a goblet cell, CC10 and plunk, which are secretory cells, including the club lineage, and then again expressing NKX2.1, indicating that they are lung-derived. We even see some of the rarer cell markers, such as FOXI1 here. But this is a new finding for us, and one that we are still actively exploring. So at this point, we know that the markers are there, and the cell types that are being expressed resemble that of the major and minor populations of the airway epithelium. We wanted to see, though, how does this ALI culture from IBC sources appear transcriptomally? Here, we performed single-cell RNA sequencing, similar to like we did above. And what we saw was the same pattern of cells, primarily keratin 5 p63 double positive basal cells, SCGB1A1 positive secretory cells, and FOXJ1 positive multiciliated cells. And again, we applied that same characterization based on the Plashert et al. Gene list, and we saw they came out as basal, secretory, and ciliated quite cleanly. We pushed forward to an integrated analysis. Now, what does this mean? Well, this means that we merged and normalized the primary HBE ALI culture and the IBC ALI culture bioinformatically. And we asked the computer to unbiasedly cluster these cells based on similarity of transcript. We used Seurat 3.1, which is the most advanced single-cell pipeline that we can at this time, and it confirmed overlap between HBEs and IBC data sets on the transcriptomic level. And this indicated to us that upon the depth of single-cell RNA sequencing, these cells appeared to be very similar in transcriptome. So now we have both markers and an unbiased transcriptome analysis. The next question is, do these cells function for CFTR research? And the answer to that is yes. And this has been one of the most difficult areas for directed differentiation to achieve. But now, due to the advances in the cultures, we're seeing reproducibility time in and time again with the IBC ALIs by electrophysiology. On the left, you see the IBC ALI. Here, we have a small benzamyl or ENaC current, but we have a very significant forskolin and 172 inhibitor currents, which indicate clean CFTR activation and inhibition. When you compare it to the HBE ALI, you see a larger benzamyl current, something that we are trying to explore why those differences exist, and a larger CFTR current. But again, the patterns are very much similar. So this indicates that the iron transport properties are typical of human airway epithelial cultures, albeit slightly reduced in their magnitude. But the real take home here is that when we generate cultures that harbor PTC variants such as CFTR G542X homozygote, we can treat them with drugs known to impact read-through like G418 and inhibit NMD such as SMIG1 and apply a corrector such as Vertex 809. When in this condition, the G542X homozygote IBC ALIs respond to forskolin and respond to CFTR inhibitor 172. And this indicates that read-through and NMD inhibition with a corrector actually can activate, albeit small amounts of CFTR that can be detected by electrophysiology from induced pluripotent ALIs. Something that is valuable for the CF field as the number of cultures produced for G542X homozygotes is insufficient for drug screening from primary sources. So just to review here, we have the right markers, we have the right transcriptome, and we have the right function by electrophysiology. Now, this by no means says that we're complete or we're done. There are still questions left to answer, but I believe we're finally making our strides in the right direction. And I'd actually like to say that it is now time for the stem cell field in CF research to start thinking about this cell-based therapy. This was done by Alyssa Miller from Jason Spence's group in a publication that was relatively recent. Here, she used a different protocol than the one I described earlier, but still nonetheless derived NKX positive progenitors. Utilizing an injury model, she instilled these into a mouse and demonstrated engraftment by focal patches of these cells, engrafting and going through multilineage differentiation. Now, this, I believe, will be the future of iPSC research for cystic fibrosis, in that we may be able to create a one-time cure event. So in conclusions, and I mean this in all earnest, it is an exciting time to be a stem cell researcher. Protocols for directed differentiation to the airway epithelia have advanced from immature lung-like cells to functionally mature epithelial cells. IBC ALI cultures are now accurately modeling the airway epithelium, with the appropriate cell fate distribution and high levels of transcriptomic similarity based on unbiased clustering techniques. Also, IBC-ALI cultures model CF as assessed by functional measurements. So, as we have been saying for many years in the directed differentiation field, there has been significant promise for CF research based on iPSCs. But now that promise is being delivered upon. I'd like to take the last moment here to be able to recognize the lab at the CFFT. And in particular, I'd like to mention Junji Liu, Alex Stufer, and Megan Simpkinson. These three make up, with myself, our STEM and Progenitor team at the CFF. I'd also like to give specific shout out to our academic collaborators, including Jason Spence at the University of Michigan, Amy Ryland at the University of Southern California, and Finn Hawkins at the Boston University School of Medicine. Together, this group has been really quite the embodiment of what science should be: resource sharing, idea generating, and collaborative from top to bottom. So with that, I appreciate your focus, and I remind you all, I'm available by chat, I'm available by message, and by email at jmahoney@cff.org. I hope you all enjoy your conference, and I wish you the best in your future research endeavors. Thank you very much.
Hello, and thanks for tuning into my talk at the 2020 meeting of the CF conference. And the title of today's talk is Genome Editing of Pluripotent Cells to Generate Cell Control Mechanisms. And I have several potential conflicts of interest with some new biotechs, but everything I talk about today was done in my academic lab, and these entities had no input. In addition, none of these companies have a FDA-approved drug on the market. So I think we're all very excited to be at the beginning of a new era in medicines in which we use living drugs, particularly cellular drugs or gene drugs, to modify the genomes of cells in the body. And because these living drugs have different pharmacokinetics and pharmacodynamics, they're likely to develop durable and treatments for patients who prior very exciting methodologies have so far been unable to address. So in the talk, I'm going to review quickly the ex vivo gene targeting system that we've developed that's highly efficient in pluripotent cells, and then review two examples of how we've used this system to engineer a safer pluripotent stem cell-based therapies. So the system we use is a combination, is based on the CRISPR-Cas9 nuclease, and it's an ex vivo system in which we purify our cell of interest, in this case, pluripotent stem cells, human pluripotent stem cells, and we put them into cycle by culturing under the appropriate conditions. We then deliver the CRISPR-Cas9 as a ribonucleoprotein complex that consists of a high-fidelity Cas9 protein complex to a synthetic guide molecule that guides the Cas9 to the site of the break. What we have found is that, and we deliver this RNP complex via electroporation. We then immediately follow the electroporation by delivering the donor template, which templates the repair of the break and allows us to create either single nucleotide mutations or insert cassettes of genes at a precise location in the genome. And we have found that this system is highly efficient in a broad variety of human primary cell types. And in another talk at this conference, I'll talk about our application to airway basal cells as a way of directly correcting mutations that cause cystic fibrosis. But for today, I will focus on how we've been using this system in pluripotent stem cells. So in 2019, Renata Martin and Kazakeda and Kyle Cromer demonstrated how effective this system was. And again, the idea is to purify our cell of interest, deliver the RNP as a ribonucleoprotein complex, and then deliver the donor template on an AAV6. And the reason we use AAV6 is that it is able to traverse the cytoplasm of cells without being detected by the type 1 interferon sensing mechanisms. And after recombination, we can get precise insertion of our gene cassette into the precise location in the genome. And this system is at least 2 logs more efficient than delivering the donor template as plasmid DNA, making the ability to screen for the right colony significantly simpler. And the system works across a wide variety of alleles, or a wide variety of genes in pluripotent stem cells, ranging between 20% to 60% targeted insertion as measured by molecular digital droplet PCR analysis. I'll note that a 20% target allele frequency, if you're targeting one allele of a gene, represents approximately 40% of the cells having at least one allele targeted. So what we want to do now that we have an efficient editing system in pluripotent cells is use this system to modify these cells in a way that we think would provide safer and more effective therapeutics. And one approach, of course, would be to genetically rescue a missing function. And many groups have shown that you can rescue CF gene mutations in pluripotent cells using gene editing. But what I'm going to talk about is how we're using another approach, which is to add new functions to the cell, sort of a variant of synthetic biology. And so the first approach I'm going to talk about is to target a specific gene to a specific locus. Actually, in this case, it won't necessarily be a safe harbor, but we know exactly what gene we're targeting into. And this just represents one of many different genetic modifications one can make using homologous recombination-based genome editing. And so this was work that was led by Volker Wiepking, a postdoc in my lab, and James Patterson, who was a graduate student in London at the time. And the idea that James had, and we worked with him to execute on, was to take the concept of auxotrophy, which is a powerful tool in niche genetics, and use the efficiency of genome editing to regenerate that auxotrophy in human cell types as a way of giving a cell control mechanism. And we've called this problem "can't live without you" because we are going to create uridine, or U, auxotrophy. So the concept is to identify a metabolic pathway and then inactivate an enzyme in that metabolic pathway such that the cell is then unable to proliferate or divide or live, and rescue the knockout of that pathway by providing a downstream nutrient. And so this is the general concept of auxotrophy. And so the pathway that we've initially identified is involved in uridine synthesis, and obviously uridine is important in nucleic acid metabolism. And we've identified a gene called uridine monophosphate synthetase, i.e. UMPS, as a target gene, because by inactivating this gene, you now make cells dependent on the supplementation of downstream uridine. You also make the cells resistant to a compound 5-FOA, which allows you to do selection. So I won't show the data on that, but it is in the paper that I just showed. So we engineered different cell types to have the UMPS double knockout using gene editing. And on the left-hand side, what you can see is that in the presence of supplemental uridine in the cell culture, the UMPS knockout cells grew almost as well as wild-type cells. But without uridine, on the right-hand side of this graph, you can see that the uridine UMPS knockout T cells grew significantly more poorly than the wild-type cells. And similarly, for human embryonic stem cells, or pluripotent cells, what you can see is that low uridine concentrations, essentially no growth of the knockout and human embryonic stem cells, but at higher uridine concentrations, there was rescue of the growth. And of course, what we found is if you added too much uridine, that could cause toxicity. And on a logarithmic scale, you can actually see the sensitivity of growth of these cells to even small changes in the amount of uridine that's provided in a culture system. So how might we use this as an in vivo safeguard for cell-based therapies that are derived from pluripotent cells? So one idea would be to, it could be a way to protect against cellular transformation. So if the product transformed in the patient, one could simply eliminate people taking the uridine and have the transformed product die. And I won't show the data on this, but this works quite well in an animal model. I want to point out that uridine triacetate is an orally bioavailable drug that is actually given and used in patients who are genetically born with knockouts in the UMPS gene. So we have an FDA-approved drug that rescues the enzymatic defect as a proof of concept. The two examples I want to give is to show how we can control the growth of an iPS-derived regenerative medicine cell product in vivo, and then how we can control the growth of an engineered T-cell product as well. So in these experiments, Volker used his UMPS knockout human embryonic stem cells, implanted them subcutaneously into an NSG mouse, fed those mice chow that contained the downstream nutrient, uridine triacetate. And then after the tumors grew to a certain size, then randomized the mice to either chow that continued to have uridine triacetate or to chow that did not have the nutrient. And what we found is that the mice that were fed chow with uridine triacetate, the teratomas continued to grow, and you can see how big they are. Whereas the mice that did not receive chow without the uridine triacetate, the teratomas stopped growing. They didn't die, which is interesting, and it gives us a different mechanism of control than some of the kill switches, which others have described, and I will describe a bit later. So we performed a similar experiment with primary human T cells, again, creating human T cells that had UMPS knockout by selecting in vitro with 5-FOA, and then again, injecting these T cells into immunodeficient mice, allowing the T cells to expand in the presence of chow with uridine triacetate, and then randomizing the mice to either chow with or without UTA. What you can see is after 2 weeks, the mice that were, after randomization, the mice that were fed chow with uridine triacetate still had a high percentage of human CD45 positive T cells in the circulation, but the mice that received chow without the uridine triacetate lost their T cells. So this gives us a mechanism to control T cell proliferation as well, but a slow mechanism, because you can see that it took 2 weeks to see the effect across all mice, whereas after 1 week, some of the mice actually had continued expansion before the T cells petered out. And so the second application is to use genome editing, not to target into a specific locus with its own promoter, as we did with the UMPS gene, but instead to harness the specific expression of a gene and knock a kill switch into that gene so you can achieve lineage-specific killing. So this was recently published in Nature Communications as a really great collaboration with Kyle Lowe here at the Stem Cell Institute at Stanford. And we developed 3 different kill switches. I'm going to just focus on 2 of them. The first one is to knock in an inducible caspase 9 into the nanog locus. And what Kyle and Renata and Jonas all did is screened for genes that were only expressed in pluripotent cells and not expressed in other cells. And nanog actually turns out to be an incredibly specific gene. And by knocking in, the idea would be that only nanog expressing nanog expressing cells would be killed when we added the small molecule dimerizer that activates the inducible caspase 9. And so again, the genetic engineering is shown at the top, where we knocked in a inducible caspase 9 linked through a 2A peptide at the C-terminus of nanog. And what we showed is in vitro, we could get over 1 million-fold cell reduction in pluripotent cells by adding less than or 1 nanomolar of the dimerizer compound. And you can see visually that essentially all the cells are killed. But importantly, When the knock-in cells are differentiated into different lineages, and then we add the dimerizer, there is no killing of the differentiated cells because these cells no longer contain NANOG. And one of the safety features of this system is that if a cell loses NANOG expression, so if it tried to turn off this kill switch, it would actually turn off its ability to be pluripotent because NANOG is an essential gene for pluripotency. So the second kill switch is what we call a kill-all switch. And instead of knocking into a specific pluripotent gene, in this case, we knocked an orthogonal inducible caspase 9 system into the actin gene. And so again, the genetic engineering is shown here, where we used the RMP86 system to knock in the orthogonal caspase 9 system, linked through a 2A peptide to the C-terminus of the actin gene. And we simultaneously had knocked in the NANOG system through the system I just described. So now the cell contains a NANOG-specific kill switch and an orthogonal actin B kill-all switch. And you can see this clone has both red, plum, and YFP, showing that it has a double knock-in. And what we can show is that the kill-all switch works extremely well. So again, we grow luciferase expressing teratomas in mice, and then randomize those mice to either the dimerizer to kill all the cells or not. And what you can see is in half a week, the teratomas are completely disappeared. And in fact, this occurs even faster than shown here. It occurs within 24 hours, as has also been shown in human beings. I've told you that the switches are orthogonal, and here's evidence of that, where when we treat the differentiated progeny of the ESCs with the NANOG-specific kill switch, we kill the pluripotent cells, but we do not affect any of the differentiated progenitors, whereas the kill-all switch that's knocked into the actin B kills all cell types, pluripotent cells and the differentiated progenitors. So this allows you then to select for which type of cell you want to kill based on which kill switch you've knocked into which locus. So how might we apply this in the future? Of course, this could be a powerful and fast safety switch if something goes awry, in contrast to the oxytrophy system, which is a little slower and perhaps more nuanced safety switch. It would be a way to eliminate pluripotent derived cell products if the therapeutic effect is completed. It would be a way of eliminating residual pluripotent cells in the cell product by activating the NANOG kill switch. And in projects that Kyle and I are very interested in pursuing in the future is by knocking into specific lineages, we hope to be able to create more specific cell populations out of heterogeneous cell populations by depleting the unwanted cell types, because the kill switch is expressed only in one lineage, but not another. So with that, I want to thank you for tuning in to my virtual presentation. I want to thank a fantastic group of postdocs, graduate students, senior scientists, and undergraduates who are in my lab with fantastic support from Ginger Luan. Great group of collaborators in my Division of Pediatric Stem Cell Transplant and Regenerative Medicine, our collaborators in the Stanford GMP facility, and collaborators both at Stanford, in academia, and biotech throughout the world, and funding from a broad range of sources. Thank you very much, and I encourage you to enter questions in the chat, and I will answer them then.
My name is Brian Davis, and I would like to welcome you today to this presentation on derivation of engraftable airway basal stem cells from iPSCs. And I would invite you to submit comments that you may have via chat, and I'll reply throughout the course of the conference. I have nothing to declare, and would like to start by talking about what is our interest in airway basal cells. And as if we look at the larger, the proximal airway, we see the pseudostratified epithelium, which has as its base the basal cells. And then above them are the abundance of ciliated cells, secretory cells, and also the rare cell ionocyte. And just shown here is an example of expression of CFTR channels on those cells that we know from single-cell RNA-seq express at least the messenger RNA. So the reason why basal cells are of interest is because it's been shown in a number of models that the basal cells are able, following injury to the airway epithelium, are able to both self-renew, that is, give rise to more copies of themselves, but also can then differentiate into ciliated, secretory, and ionocyte cells. So there's a possible role of these cells that they may be useful in regenerative medicine, that if they were to be delivered to the airway, that they could then result in the repopulation of cells derived from them. So this is just showing here sort of the normal airway epithelium with the channels and the ciliary function. In the case of CF, where you do not have channels, no ciliary, minimal ciliary function and thick mucus. And the idea is to use those airway basal cells to basically correct them and then to deliver them into the airway so that they would essentially seed the emergence, the differentiation of the various types of specialized cells that I spoke about in a sense self-renewing population, that they could then do this hopefully throughout the life of the patient, restoring the CFTR function. So one is can think about either using primary airway basal cells that are expanded ex vivo, and there's work being done on that. But what I like to focus today is on the possible use of induced pluripotent stem cells to derive that airway basal cell population. So let's first talk about what might be the biological or functional properties that we're looking at of the airway basal cells that we'd have to recapitulate in any iPSC derived airway basal cell. So transcriptome been characterized by single-cell RNA-seq, self-renewal and multipotent differentiation. I'll talk further about the bronchosphere assay, as well as the ability in air-liquid interface culture to derive an airway epithelium. Also, the airway epithelium can be used in Ussing assays for measurements of CFTR activity, and finally in vivo engraftability. And in this case, if we're working with human cells, thus far the ability to engraft these cells into the airways of mice, for example, is, although it's developing, is still rather limited. So we, in fact, have been using a tracheal xenograft model that I'll talk about in just a moment. So if I look here, again, this is with airway basal cells, primary derived from the lung, that if we take, for example, an air-liquid interface culture to assay multipotent differentiation, you see the pseudostratified epithelium, and you see the emergence essentially of cells that are expressing basal cell markers, in this case keratin 5, ciliated markers, acetylated tubulin, various secretory markers like the mucins or secretoglobulin, and then finally FOXI1 expression characteristic of the ionocytes. And just showing over here is the ability, in this case, of wild type or non-CF. You see a nice forskolin-induced response in the Ussing assay that's inhibitable by a CFTR-specific inhibitor. The tracheal xenograft involves taking these airway basal cells, for example, from human, seeding them into a denuded rat trachea and engrafting it into the back of a nude mouse, and one basically retains open-ended tubing so that the lumen is continually exposed to air. And what you see after 3 to 5 weeks is essentially a pseudostratified epithelium that again has the basal, the ciliated, the secretory, and the ionocyte markers. So this is in fact the assay that we're going to use. We're under the normal sort of providing of factors that come through the circulating blood of the animal and the airway exposure. One is able to derive an airway epithelium that's exclusively human cells. So if we think about how we may use iPSCs for this purpose using an airway basal cell protocol, is one starts with either skin or blood from a CF patient. One has the mutant iPSC cells through reprogramming. Then you can use these airway basal cells, either in vitro to essentially form these ALI cultures that could be used for testing response to modulators, or one can edit or correct the CFTR mutation directly in the iPSCs, the CFTR gene, and then derive the basal cells that could then serve this role of essentially restoring CFTR function to the CF airway. So at least that's what our paradigm is in terms of what we wish to do in developing this airway basal cell protocol. So let me describe then the dual reporter approach that we used. We wanted to have fluorescent proteins that would become activated and expressed at specific hallmark key steps in the development process that would allow us to both identify the emergence of either lung progenitors or airway progenitors as they were arriving culture, as well as to then further develop or optimize the protocol. So we published already 3 years ago the ability to use an NKX 2.1 GFP reporter to identify lung progenitors as they emerge in vitro differentiation from marked iPSCs. And what we have done now more recently is to introduce a second reporter, TD tomato, into the P63 or the TP63 locus. P63 is a hallmark transcription factor associated with basal cells, so allows us to identify these airway progenitors. And then we've subsequently used a surface marker, NGFR, nerve growth factor receptor, to then be able to identify cells as they mature from airway progenitors into the airway basal cells. And this just shows our protocol for generating these NKX 2.1 positive lung progenitors, and is shown here. Over time, one sees the emergence of those NKX 2.1 positive lung progenitors here. And then by doing essentially a Wnt withdrawal, this is a protocol developed by Macaulay et al. In 2017, what she was able to show is essentially Wnt withdrawal leads to airway differentiation, and we now have in 3D these spheroids that have cells that now acquire P63 expression, where a good percentage of the cells are now expressing both NKX 2.1 GFP and TD tomato, and that's just shown here in this upper right quadrant, the emergence of these cells that are double positive. And just shown over here is a live cell imaging where you can see the co-expression of basal cells that are expressing both the GFP and the TD tomato. I should be careful here, at least they're airway progenitors at this time. So what we now have looked at is if we take these organoids, we sort out the double positive cells, and we maintain them in our essentially airway media, although we can maintain and expand the presence of these double positive cells, that is TD tomato positive, GFP positive cells, we do not see the emergence of the mature basal cell marker NGFR on the cell surface. But instead, if we take these same cells and put them into what we call basal cell media, which is essentially a basal cell expansion media that contains a dual SMAD inhibition that was proposed by Hongmei Mo a couple of years ago for expanding basal cells together with ROCK inhibitor, we now see not only the maintenance of the double positivity, but now all the robust emergence of NGFR expression. This is shown here in the blue is how rapidly is the acquisition of the NGFR expression by a vast majority of the double positive cells. And just showing here, if we maintain the cells exclusively in the airway media, we do not see that. So the question is, we now have these triple positive cells that we're calling IBCs, or induced basal cells. Are they in fact equivalent to adult airway basal cells? So we're going to basically use the duck analogy. We're going to test and evaluate the transcriptome as well as several of these biological or functional properties that I spoke about earlier as being characteristic of the airway basal cells. Let's start with transcriptome. So this is showing here in turquoise the cells that were maintained in the basal cell media, as opposed to in pink the airway media. And what you can see here is when we apply a basal cell signature, that a certain high frequency of the cells now have that signature. If we then analyze by single cell RNA-seq, and we look again at these turquoise cells here, and we look across a variety of sort of developmental stages. And we can also look at adult. We can see that it's primarily these adult basal cells that have a transcriptional profile that highly lights up with these basal cells, and in fact is the best fit for the cells that were maintained and expanded in the basal cell media here. If we look at function, I'm going to start with the bronchosphere assay, where essentially these cells that are now double positive and also have induced NGFR because of the maintenance in basal cell media, those cells are now seeded as single cells in matrigel and form over a period of days these well-defined spheroids that, when they're maintained in basal cell media, are essentially these undifferentiated basal cells, retain expression of NKX 2.1 and TP63. They expand and robustly proliferate, but still retain by and large this undifferentiated phenotype. But then when they're put into this air-liquid interface differentiating media, you now see, in addition to the basal cells around the outside, you see the emergence of cells, differentiated cells, either ciliated cells or secretory cells present. And this ability to start with one cell and give rise to a full spheroid demonstrates the self-renewal capability, and the emergence of the secretory and ciliated cells demonstrates the differentiation. We can also demonstrate this multipotent differentiation in the air-liquid interface culture, where you see over here, basically when there's an airlift performed, you see the well-developed airway epithelium that has all the hallmark basal cell secretory and ciliated cell markers. Here you clearly see the secretory, or sorry, the cilia in close-up, and then also with the staining for ZO3 that they have very good tight junction formation. So what I've described thus far is the protocol for development of these basal cells coming from this double reporter line, but obviously we don't wish to introduce these reporters into every cell that we wish, iPSC line that we wish to study. So we've basically applied this methodology and have developed an approach that, using surface markers, in this case, either sorting at the day 15 stage, for example, for NKX 2.1 positive cells, or enriched by using CD47 high, CD26 negative, or using the CPM marker of Dr. Goto. We're able to enrich for those cells. We then put them into the airway media, then into the basal cell media, and then we're able to sort for NGFR positive, EpCAM positive cells, and similarly develop this airway into air-liquid interface culture. And just showing here is that the cells, once they differentiate, retain their CFTR function that's appropriate for their genotype. The mutant homozygous delta 508 cells give rise to negligible current. If we correct by editing one of or both of the alleles, we see a very beautiful CFTR inhibitable current. And similarly, if we take either rare genotypes or unknown, in this case, we're taking G551D, we see that in fact it recapitulates the anticipated VX770 responsive current. So that demonstrates sort of the function sort of in vitro. What we're showing now here is if we take the double positive cells and also sort for the NGFR positive and the EpCAM positive, this is using the double reporter line, and use that to develop the tracheal xenografts, you see the clear development of the airway epithelium by H&E. And then if we now look for the basal cell, secretory, or ciliated markers, we now see them all present in that well-developed airway epithelium. And just showing here by staining for GFP and DD tomato, we're able to demonstrate that in fact the cells giving rise to this airway epithelium are from our human IBCs and not from mouse cells. So essentially, I think we've shown here that we sort of checked off the boxes for these different biological or functional characteristics. But is it really satisfying all the criteria we expect? And I would point out that when we took our IBCs and put them into the air-liquid interface culture, we then analyzed by single-cell RNA-seq. You see the basal cells, you see the secretory, the multiciliated cells, but lacking were the ionocytes. And this really raised a question for us whether our protocol was in fact able to give rise to the ionocytes in addition to the other cell types that we expected. And I'm very pleased to say that presented at this meeting that Dr. Ruby Wang from Boston Children's is presenting a poster in which now she demonstrates using this particular protocol. She's incorporated some additional sorts for NGFR along the way and a little bit further expansion. And in so doing, she demonstrates now that the derived airway epithelium using this protocol, in fact, does have ionocytes present, as is seen in that from primary airway epithelium. So in conclusion, I believe that we can sort of check off all the boxes, at least of these cell types that we believe are of interest for cystic fibrosis and eventual therapeutic use. So I'd like to just finish by acknowledging both my colleagues at UT Health, especially Dr. Shingo Suzuki, who really played a key role in spearheading this work, working closely with Dr. Finn Hawkins, as well as Daryl Cotton at Boston University. We've had great collaborations with colleagues at Emory, University of Michigan, Cedars-Sinai, Boston Children's Hospital, and we're very, very grateful for the funding that we've received to permit the performance of this work. In closing, I'd just like to direct your attention to 4 posters that either use this very protocol or even further developments, optimizations for maybe those lines that are more difficult to turn into airway epithelium. So I'd like to direct your attention to Christina Barilla from my laboratory, Andres Rob from Emory, and Shingo Suzuki, who are sort of highlighting the use of that protocol. And then finally, I've already mentioned Ruby Wang and her work on derivation of ionocytes. So thank you very much for your attention, and please feel free to direct any questions or comments to me by chat. Thank you.
Hello, everybody. I want to thank the NACFC, Dr. Ryan, and Dr. Hawkins for giving me the opportunity to present our work at this meeting. I'm a thoracic lung transplant surgeon at the University of Iowa, and my laboratory focuses on the role of airway epithelial stem cells in lung transplantation. In my talk today, hopefully, I will try to outline how we got interested in exploring a potential therapeutic role for stem cells for lung disease and highlight some of our work and challenges. I have no financial disclosures. To give you a background on the major stem cell types in the lung, lung is a complex organ with several trophic units, each with a unique function, cell type composition, and progenitor cell niche. The large conducting airways are pseudostratified, which become cuboidal in the bronchioles and largely squamous in the alveoli where the gas exchange occurs. Each of these tissues are able to regenerate after injury and possess specific cell types that serve as tissue-specific stem cells for that trophic unit. In the cartilaginous airway, submucosal glandular ductal basal cells have been identified as progenitor cells for the surface airway epithelium. And recently, our group has identified myoepithelial cells in the submucosal gland that have regenerative properties. What defines a cell as a stem cell? Ideally, these cells have the ability to self-renew and can differentiate into all cell types within a given biologic unit. Stem cells typically reside in a supportive stem cell niche that maintain quiescence and multipotency when needed. Upon receiving a signal to repair, the stem cell typically undergoes an asymmetric cell division, whereby it generates self-renewal in a transit amplifying cell that can proliferate and differentiate to maintain homeostasis. Other important mechanisms of cellular regeneration in the lung include facultative stem cells. In this context, facultative stem cell refers to the ability of a committed differentiated cell to de-differentiate it into the stem cell that gave rise to it. 2 examples in the lung include the club cells that can de-differentiate into a basal cell type that can repair injury, and alveolar type 1 cells that can de-differentiate into type 2 cells and can generate alveoli. One of the most well-characterized progenitor cells of the proximal airway is the basal cell. These cells have the ability to generate all cell types of the surface airway epithelium in response to injury and maintain homeostasis. In addition, the submucosal gland also contains progenitor cells called the ductal basal cells. The myoepithelial cells that are present around the submucosal glands play an important role in embryogenesis and glandular formation. And recently, our group and Purushottam Rao Tata's group at Duke University have also shown that these cells possess regenerative properties in the adult animals. Here's an electron micrographic picture of the myoepithelial cells around the submucosal gland, shown in red. Tom Lynch from our group used lineage tracing for these myoepithelial cells based on expression of alpha smooth muscle actin 2 and myosin heavy chain 11. He crossed these Cree drivers to a Cree reporter to obtain reporter mice. Upon induction with tamoxifen, these mice result in an MEC labeling efficiency greater than 80%. Thus, these mice could be used for lineage tracing of the myoepithelial cells. Using this mice in an injury model with naphthalene and sulfur dioxide, Tom showed that these myoepithelial cells are able to generate all cell types in the surface area of epithelium. As you can see, these GFP positive cells that are ciliated cells, club cells, goblet cells, and non-ciliated columnar cells following injury that arise from these myoepithelial cells. He also showed that these cells are capable of generating not only surface area epithelium, but also glandular epithelium, including mucous cells, serous cells, and ductal basal cells. Now in mice, the submucosal glands are present only in the first and second tracheal ring, whereas in humans and in ferrets, the submucosal glands are present throughout the entire cartilaginous airways. And therefore, we use the ferret as the model for our orthotopic lung transplant. My laboratory has developed their orthotopic left lower lobe transplant model in the ferret, which develops obliterative bronchiolitis similar to human allografts. Obliterative bronchiolitis is a pathologic condition that develops in human allografts wherein the distal airways are completely occluded with scar formation and collagen deposition and results in decline in function of the lungs. The ferret model develops OB lesions exactly similar to the human allografts. Using this model of OB, we have shown that there is progressive destruction of the submucosal glandular niche in the airways of the ferret allografts as OB develops. You can see here the submucosal gland destroyed and replaced by inflammatory infiltrate as OB progresses in the allograft. When we quantified the glandular clusters, there was a progressive decline in the OB allograft. This destruction was also observed in human allografts that's shown here. You can see a normal human airway showing nice glandular structures, whereas an OB allograft showing complete destruction of the glandular structure. Quantification revealed again similar data to the ferret data with decline in the number of submucosal gland clusters in human OB allografts. When we looked at the basal cells in the surface airway of the allograft, both in the large and in the small airways, there was a progressive depletion of the K5 positive, P63 positive basal cells in the large and small airways of both human and ferret allografts. When we looked at the phenotypic markers of the basal cell, we noted that while the number of the K5 positive and P63 positive cells declined in the allograft, there was a corresponding increase in the number of K14 positive cells as OB developed in the allografts. This was seen in both in the ferret and the human allograft that developed OB. Based on this data, we asked the question if whether stem cells can be used as a therapeutic tool to prevent or slow down progression of the obliterative bronchiolitis in the transplanted lung. When we started to address the hypothesis of stem cell therapy, we began to realize that there are several challenges that we need to overcome, some of which are listed here. One challenge that we need to address is whether recipient-derived stem cells could repopulate the allograft, as these cells would have an immunological advantage over the donor-derived stem cells. Another important question we needed to address was the ideal cell type for therapy, whether it be surface area basal cell, whether it be myoepithelial cells from the submucosal gland, or even iPSC-derived progenitor cells. Amy Ryan's lab has developed iPSC-derived progenitor cells that can differentiate into airway epithelial cells in both human and ferret cell types. Other questions that we need to address are the way to deliver these stem cells in the airways and the timing and frequency of the delivery. What we really needed was the ability to track these cells in order to test our hypotheses, and the Engelhardt laboratory has developed the Rosa TG ferret in which all the cells are TD tomato positive, and on induction with tamoxifen, they can turn GFP positive. This animal allows us to track these stem cells in a wild type airway. We first needed to develop an injury model where we could try out cellular therapy. So we performed a proximal injury model using a mechanical brush endoscopically, as shown in the video here, where we injured the ferret airway using a bronchoscope and a transcatheter brush that injures the airway epithelium mechanically. When we looked at these airways histologically, there was complete depletion of the surface airway epithelium, and there is recruitment of K5 positive and K14 cells in the airways to repair these injured area. Using this model, we wanted to try out our cellular therapy approach. So we first started by expanding Rosa TG ferret basal cells. So we took a Rosa TG ferret, we did a tracheal brush biopsy, and expanded the Rosa TG basal cells using rock inhibitors for about 2 weeks. And 2 weeks later, we take a wild type ferret and we mechanically injure the trachea as shown in the video in the prior slide. And then we deliver Rosa TG basal cells for repair in the area using bronchoscopic delivery. And we checked at several time points to see if there was engraftment of these Rosa TG cells in the injured area with and without immunosuppression. When we looked at 3 days without immunosuppression, we noted that the Rosa TG basal cells were engrafted over the injured area, and they maintained basal cell markers as shown by K5 positive cells and K8 positive cells, which is a marker for luminal fate commitment. Here again is another slide showing that these cells are TD tomato positive and have K5 and K14 positive cells. So these cells maintain basal cell markers at 3 days following delivery. When we used immunosuppressive drug, we noted that there was a significantly less engraftment of the TD tomato positive basal cells in the injured area, raising the question whether the immunosuppressive drugs affects proliferation, adhesion, or affects the inflammatory microenvironment in the area. At 7 days with immunosuppression, we continue to see small pockets of engrafted TD tomato positive basal cells. And they begin to show signs of differentiation at this point. They are keratinate positive, TD tomato positive. And when we look at differentiation markers, we can see that some of the cells are positive for MOC5AC and MOC5B, suggesting a secretory cell phenotype differentiation. We also saw some cells that were alpha tubulin positive and TD tomato positive, suggesting a differentiation into a ciliated cell type. We next wanted to move to a transplant model and see if we could repopulate an allograft with autologous cells that would be recipient derived. We decided to take baby steps and not try this immediately in a lung transplant model. So we first attempted to see if we could recellularize a decellularized tracheal scaffold with the TD tomato positive basal cells. Now this work is largely done by Dr. Albert Pai, a resident physician in my laboratory. To achieve this, Albert first designed a cost-effective reusable bioreactor as shown here in this image. He used a 50 ml conical tube cap to accommodate silastic tubing and lures to achieve a watertight configuration. The tubing components can be autoclaved while the caps are sterilized in an alcohol bath. Using this, we exposed a trachea based perfusion protocol adopted from Dr. Kruby and Dr. Tom Wattles' group at University of Toronto, delivered by this peristaltic pump. Submersion of the circuit into a basic media preserves the adventitial component of the trachea, whereas the luminal surface is decellularized. The inset image here shows the normal trachea under immunofluorescence, which shows basal cells marked in green and nuclear marked in blue. In contrast here, this image shows a section of the tracheal graft that was treated with the decellularization protocol. There is complete absence of well-organized nuclei and keratin 5 positive cells superficial to the basal lamina. And please appreciate the preservation of the submucosal glands within the submucosa as marked by the yellow asterisks. To initiate the recellularization process, we isolate and expand tomato-positive tracheal basal cells from a transgenic Rosa TG ferret, and then a centrifugation method of recellularization adapted from Sayo's group in Japan was used for the initial seeding of these cells on the decellularized trachea. After 24 hours of the Sayo's method, we reattach the seeded trachea to the bioreactor as shown here. And expansion media is regularly perfused through the lumen by syringe aspiration as shown here. The adventitial basic media supports the chondrocytes and selects against the basal cells that have inevitably seeded on the tracheal, external trachea. 2 days after this expansion, we obtain a section of this trachea and we show that there is engraftment of the TD tomato positive cells, which have formed a monolayer on the tracheal surface. By day 5 in vitro, the basal cells begin to proliferate and colonize the submucosal gland as shown here by the yellow asterisks. To encourage differentiation, Albert modified the bioreactor to establish an air-liquid interface, as shown in this picture. The adventitial aspect is submerged in the differentiating media, and the lumen is exposed to incubator air conditions. This condition allows for differentiation, and at 12 days, we can see keratin 8 marker positive cells on the surface airway. We next transplanted the 10 to 12 ring segment of trachea into a wild-type animal. This is a tracheal graft that was decellularized and recellularized with TD tomato positive basal cells, and then transplanted into a wild-type trachea after being in bioreactor for 3 days. We use the muscles in the neck and VEGF to promote vascularization of this tracheal graft. 3 days after the transplantation, the grafts show preserved epithelium and submucosal gland colonization by the Rosa TG ferret basal cells. At day 7, the cells continue to persist. However, there is no evidence of differentiation yet. When we look at basal cell markers, these cells continue to possess K5, K14 positive cells. And they also show K8 positive differentiation, suggesting a luminal fate commitment at this point. So in conclusion, we show here a cost-effective bioreactor that is capable of decellularizing and supporting a recellularized ferret tracheal graft. Airway basal cells cultured in the bioreactor have a potential to engraft and potential to differentiate. And orthotopic transplants performed using these seeded tracheas are able to maintain the epithelium in vivo and maintain basal cell markers at early time points. Future directions include optimizing engraftment techniques and exploring different cell types, including iPSC basal cells or myoepithelial cells, which have shown to be a reserved stem cell within the submucosal gland niche. We will also assess the long-term survival and differentiation of these cells in the grafts, and also evaluate the repair potential of these engrafted airway cells by inducing injury at later time point after the transplant. Finally, we also want to observe the kinetics of the reepithelialization between the native and transplanted cells. That is all I have. I want to thank Dr. Engelhardt, my mentor. The transplant OB work is mainly work of Dr. Tony Swatek. The myoepithelial cell work is done by Tom Lynch, who's a postdoc in my lab. And the tracheal transplant work of decellularization and recellularization is done by Dr. Albert Pai, who's a resident physician in my lab. I thank you very much again for the opportunity, and I'll take any questions at this point.